So after that, you can just continue it.
Okay.
Yes, wait for 30 minutes only.
Okay.
30 seconds, sorry, 30 seconds.
30 seconds. 30 seconds.
Yes, sir,
YouTube,
yes.
YouTube will be started. Yes, you can start.
Okay, sir.
Sure, sir, sure, sir. Sir, I'll start with your with your introduction first. So,
yes,
yeah.
yes,
So good afternoon, everyone, and welcome to this beautiful lecture series of Sunday Talkies on researchers talking from abroad. And I welcome you all, and I'm Dr. Nitika Thakur, and working as assistant professor in the Department of Biotechnology, Shulin University, Himachal Pradesh. So today we have a very beautiful presentation and a very relevant topic related to microbiology, that is antifungal and antimicrobial activities of G-90 coelomic fluid of earthworms and metabolites from mutualistic microbes with earthworm by Mr. Godeta, and he's serving as a type of lecturer in various type of institutes and colleges in Ethiopia. Before we move on to his presentation, I would like to request our president, Professor Arvind Deshmukh, sir, to kindly share the vision and mission of Microbiology Society India. Over to you, sir.
Thank you. Thank you, Nikita. This Microbiologist Society is working to serve the humanity. And many active young teachers and old teachers are working in this organization. I am very happy today. A scientist from Ethiopia, he is delivering lecture and on antifungal and antimicrobial activities. So it is my duty as a president of Microbiology Society to wish all the best to the convener and to the speaker as well as to the attendee. Thank you. Thank you very much.
Okay. I would like to salute and extend my warm greeting to all faculty, scientists, professors, and students, and welcome to be my audience of international presentation. International presentation on antifungal and antimicrobial activity of G-90, coelomic fluid of earthworms, and metabolite from mutualistic microbes with earthworms. Kasaun Gudata, research scholar, School of Biological and Environmental Science, Shulin University. Introduction. Earthworms are invertebrate animals belonging to phylum Annelida. They are coelomates with intersegmental septa separate the coelom into tiny compartments filled with coelomic fluid. Due to their habitat, soil environment full of pathogens, they have developed a mysterious defense mechanism that can defend soil pathogens. The antimicrobial activity that prevents them from soil pathogen is achieved by bioactive molecules in their tissue and coelomic fluid. The metabolite that's secreted from bacteria, making mutualistic relation with earthworm also, assists them to resist the effect of pathogen in the soil. Secretion in the biology of coelomic fluid. Coelomic fluid is a yellowish colored alkaline biofluid composed of watery matrix, plasma, specific protein, enzymes, salt, and at least all coelomocytes, namely amoebocytes, mucocytes, circular cell, and chlorangulin cell. Coelomic fluid contain enzymes such as protease, metalloenzyme, lysozyme, fibrinolytic enzyme, chitolytic enzyme, and antimicrobial protein. They have agglutinating, proteolytic, hemolytic, mitogenic, antipyretic, tumorytic, antibacterial, and antifungal activities. Each compartment of coelomic fluid, coelomic cavity has a paired nephridia and a single dorsal pore through which coelomic fluid is ejected. Coelomic fluid is secreted whenever the worm irritated or while they are moving to moisten and lubricate their burrow and thereby defend the pathogen in the soil environment. Secretion of coelomic fluid. Earthworms possess very poor defense mechanism, and instead they ooze out coelomic fluid from their dorsal pore in response to unfavorable environmental conditions. This organism is coelomate organisms that possess coelomic cavity and coelomic fluid. Coelomic cavity is found in between the dorsal endoderm and the ectoderm. It is separate from gastrointestinal cavity, and they have many pores. Many of the pores are concerned with the reproductive function, and some of the pores are actually dorsal pores that are found on the dorsal side, and it is used to secrete coelomic fluid. Coelomic fluid extraction. 6 methods of coelomic fluid extraction are there. It is based on irritating the animals to cause them to secrete the coelomic fluid. One is electric shock method called water shock method, warm water shock method, heat shock method, and alcohol irritation method. And the other one is a puncturing method, which is different from the irritation method. This is simply inserting needle by the syringe into their coelomic cavity and then suck coelomic fluid from the body of the earthworms for the experimental purposes. Preparation of tissue extract or G-90. It is also, another product is G-90. I have told you that two different products are there. One is coelomic fluid and the other one is G-90. G-90 is not extracted, but it is prepared. It can be also named as G-90 tissue homogeneous tissue extract or earthworm paste. It is made by mincing the entire body of earthworms. First stated by the Risenjak and his colleague during 1992. Antimicrobial activity in the earthworms. The lifestyle and the habitat of earthworms expose them to survive in an environment where pathogen abundance is present in the soil. And they adapt to defend themselves against soil pathogen by variety of humoral and cellular methods. They developed the different variety of defense mechanism, and hence their defense is called mosaic defense mechanism, because they have, there are many different pathogenic microorganisms are there in the soil. So they have different antimicrobial activity that is achieved by different bioactive molecules that are found in coelomic fluid and in tissue homogeneous. So these are the bioactive compounds that are found in coelomic fluid and they are tissue homogeneous. The first one is, the column is there, bioactive molecule, molecular weights of the bioactive molecule, mechanism of defense, and this is a source, whether it is coelomic fluid or G-90 or skin secretion. So the first one is lysozyme. This is a bioactive molecule that is found in earthworm, first identified from the Eisenia fetida with a molecular weight of 39 kAd. Its mechanism of action is extracellular lysis and act to recognize between microbes. The second bioactive molecule is lumbresin 1 and lumbresin 1, 6-34, first identified from the lumbresin luberus. Lumbresin luberus is earthworm species with 7.2 kAd, act against the broad spectrum of microorganisms without hemolytic activities. The other one is lysanin, and is identified from the Eisenia fetida with 33 kAd and 41 kAd. Lyse the cell by acting on the cell wall of both gram-negative and gram-positive and gram-negative bacteria, and it is spore forming. The other bioactive molecule is coelomic cytolytic factor. It is another bioactive molecule that was first identified from the Eisenia fetida with a molecular weight of 41 kAd. Its mechanism of action is lysis bacteria cell, and that is spore forming. Another bioactive molecule that is found in earthworm is lumbresin PG. This lumbresin PG was first identified from Ferretum magularium with molecular weight not identified. It is short peptide with 39 amino acid residue. It acts as primary defense mechanism against fungi, bacteria, and viruses. And the other one is Eisenia-4. Eisenia-4 was first identified from Eisenia fetida, species of earthworm, with 38.6 kAd. Its mechanism of action is cytotoxin in the pore forming. The other bioactive molecule is fetidine, was first identified from Eisenia fetida with a molecular weight of 40 and 45 kAd, with a mechanism of action of hemolytic and pore forming. And the other bioactive molecule that was found in the coelomic fluid in the coelomic fluid of earthworm was the OEP 3121, and this is the name which is given for this unique bioactive molecule first identified from Yesenia foetida with a short peptide with a mechanism of action of lysing the cell. And the other one is bacteriostatic. Bacteriostatic is another name of the bioactive molecule that is found in the coelomic fluid of earthworms, first identified from Yesenia foetida with 20, 40, and 45 KD, lysing the bacterial cell and prevent the bacteria from growth by pore forming. Most of them was identified from coelomic fluid or extracted from coelomic fluid, except that of the Lumbricin PG that was first identified from Ferretimaglurium, extracted from their skin secretion because it is act as a primary defense mechanism. That's why it is found in the skin secretion of earthworms. Antifungal activity of coelomic fluid of earthworms. Coelomic fluid, the efficiency of coelomic fluid was experimented by Rajesh and his colleague on the fungal group, on the species of earthworm, Eugenia, by using some of the fungal pathogen, like for example, Persilla dahalia, Aspergillus flavus, Rhizoctonia saloni, and Fusarium oxyporium. And this actually this name is this code is microbial type culture collection gene bank located in Chandigarh. It was purchased from the place where it is cultured in Chandigarh. In the presence of coelomic fluid, in the absence of coelomic fluid, they experimented, grows with coelomocytes, that is experimental, and grows with control in the presence of coelomocytes. Their growth is greatly suppressed in the absence of coelomic fluid. They grow vegetatively. So indicating that the coelomic fluid contain some of the bioactive compound that can suppress the growth of fungal pathogen. So antifungal activity of coelomic fluid from extracted from different species of earthworms. They extracted different coelomic fluid from different species of earthworms, genus Eugenia, Yesenia foetida, genus Eugenia, and Lumbris lubicus. And the fungal pathogen was also, used. The fungal pathogen that was used, Rhizoctonia saloni, Fusarium oxyporium, Aspergillus niger, Rhizoctonia saloni, and Fusarium graminearum. And different scholars identified that the coelomic fluid that were extracted from different species of earthworms suppress greatly the growth of fungal pathogen. Antifungal activity of G-90. G-90, as I have explained earlier, it is prepared from the entire body of earthworm by mixing the entire body of earthworms, and it was also detected or evaluated. It is a fungal activity. The fungal activity was actually evaluated by using different volume of G-90 or tissue homogenate at 50 microliter and 100 microliter. At 100 microliter, the zone of inhibition of fungal growth in millimeter was greater at around 13.3, and this is during when the Aspergillus flavor is used, 15 millimeter, and when the Penicillium notatum is used, 14, indicating that this is also the bioactive molecule that is able to be found in the tissue homogenates. And this is also another experiment that was performed by a different scholar, and they use different volume of earthworm test, starting from 25 ml, 15 ml, 100 ml, 150 ml, and 250 ml by using fluconazole as a control group. Fluconazole is the main or the known antifungal agent that is used by the pharmaceutical agents. And look, at 150 and 200 ml, it is almost equivalent or equal to the antifungal activity of fluconazole. At 15 12.8, and the 200 11.6, and fluconazole is its efficiency of fluconazole at 20 ml is 12.4, and 12.2, 14.8, 16.4. Here is 13, and then 30.8 and 14.6, 12.12, and the 13.4, 30.8. Almost when the higher volume of the paste, earthworm paste used, it is equivalent or equal to the known antifungal agent fluconazole. Antifungal activity of metabolite from mutualistic bacteria with earthworms. I have tried to explain earlier that the earthworms are able to make mutualistic relationship with microorganisms. So one of the microorganisms that are able to make mutualistic relationship with earthworm is Pseudomonas. Pseudomonas, the species of Pseudomonas or different species of Pseudomonas can secrete different metabolites. So the metabolites that are secreted from different species of Pseudomonas can suppress the fungal pathogen to fight against or to suppress the fungal diseases. So, and these are the metabolites that are secreted from different species of Pseudomonas. Pseudomonas are the known species of bacteria that can make mutualistic relationship with earthworms, and then they, when they are making mutualistic relationship with earthworms, their growth is greater, and around or along with earthworms, they grow very well. So they can, secrete such kinds of metabolites. These metabolites are very important to defend the earthworms from any pathogenic microorganism, especially fungal pathogen. So its application for use. The synergetic effect of all these bioactive compounds in coelomic fluid, skin secretion, and the metabolites from bacteria, making the soil environment free of any pathogen, phytopathogen. Therefore, farmer can easily prepare vermiwash and vermicompost and add it into their farmland to suppress any pathogen, whether it is bacteria pathogen or fungal pathogen in the soil. Preparation of vermicompost and vermiwash. Vermiwash or vermicompost can be prepared easily with a cheap cost by the farmer. So these are the way of simple design of preparation of vermicompost or vermiwash. The first one is, the first layer decomposed organic matter, a cow dung is there, and the third one from top to down, earthworm soil with soil and sand is there, and on the fifth layer from top to down, stone and brick, and then finally after 35 days or maybe 35 up to 14 days, we can collect vermiwash and we can apply this vermiwash on farmland to suppress any of the pathogenic microorganisms. And the rest of the decomposed organic matter that are solid can also be used as a vermicompost and then applied on the farmland. It is actually hitting two birds by one stone, which means it can also increase productivity by increasing the fertility as well by suppressing any of pathogenic microorganisms that are found in the soil. Future direction in the soil where earthworms are present, any pathogen cannot survive due to the antimicrobial, antifungal activity of bioactive compound in the coelomic fluid and the skin secretion. Therefore, the importance of earthworm to suppress the effect of soil pathogen should be advocated to the farmer by agronomists, researchers, and other concerned bodies so that they can prepare vermiwash or vermicompost for use by inoculating the worms and propagate them in their farmland and other land use system. Conclusion, the immunity of earthworm is achieved by a variety of both humoral and cellular immunity that developed as a result of evolutionary process of adaptation. They defend the pathogenic microorganisms through potent bioactive compound in their coelomic fluid tissue, skin secretion, and metabolite from decomposer bacteria. This product should be applied by the farmer and agronomist to suppress plant disease and increase productivity of crops. References. The reference that are used in this presentation are The paper that were published recently, the first one is vermiwash, an agent of disease and pest control in the soil. And the second one is vermicompost, and it is derivative against phytopathogenic fungi in the soil. Thank you to the audience of my presentation. With this, I completed my presentation. Thank you.
Thank you so much, sir. It was wonderful hearing to you and hearing your research experiences, the real research experiences. And it was awesome, viewing your type of publication, research publications, basically. That was generally the input of your and the output of your research work. And the house is now open for discussion and your queries, please.
Yes.
If anybody is having any type of queries.
Yeah.
Yes, ma'am.
I'm Dr. Sujita.
Welcome, ma'am.
Yeah, good afternoon, good day, sir. Actually, I have a question. I'm working on antibiotic resistance and antifungal products. Whatever your work is there, it's really wonderful. And it's if its outcome is commercialized, it will be a boon for farmers. In the last application, you said that it can be used for plant sprays or something. So,
Yes.
you tested the cytotoxic activity of the fluids?
Yeah. Look, it is already published, and many researchers already proved that the bioactive compounds are there that I have listed. You know, there are bioactive compounds that are found in the tissue homogenate and coelomic fluid. Tissue homogenate means is a main thing of the whole body of earthworms. In that, there are bioactive compounds. Look, and these are the bioactive compounds that are found in the coelomic fluid. Lysozyme, lumbresin, lysine, coelomic cytolytic factor, all these, lumbresin PG, infetidine, isinaphore, all these are found on in the vermiwash. Okay. And the other one is, the metabolites that are secreted from the bacteria, making child-sick relationship also there. So, the synergetic effect of the bioactive compound that are found in the coelomic fluid, skin secretion, and the metabolites are found in the both vermicompost and vermiwash. So when we are preparing vermicompost and vermiwash and apply into our farmland, it can use for two purposes. One, it increase productivity. Vermicompost, has got many nutrients in it because it is a result of decomposition of large and very complex food organic matter into very simplest. So in that regard, it can enhance the productivity. And the other one is the bioactive compound that are found in coelomic fluid, skin secretion, metabolites that are secreted from decomposed bacteria that can actually they are decomposer. Bacteria making characteristic relationship with also able to suppress any of pathogenic microorganisms in the soil. So it has two purposes: one to increase productivity, and the other one to suppress or to inhibit any of the activity of pathogenic microorganisms in the soil.
Yeah.
Yes.
Yeah. Thank you.
Yeah. Okay. Thank
you. I have one question.
Yes, sir. Welcome, Saranjit, sir.
Thank you so much, Nitika. So, can I ask one question to him?
Yes, sir. Sure.
Okay. It was a wonderful talk, very nice talk. And I was happy that it is on vermiculture. Now, what are the antibacterial compounds which are made by the earthworm?
What are antibacterial agents?
So what are the antibacterial? Yeah.
Yeah. Yes. Actually, it needs further investigation. They are protein. Okay, they are protein bioactive substances. For example, if you see lumbresin PG, it is a short peptide with 59 amino acids. And most of them, GP, for example, in glycoprotein, GP90, G90 is termed because of the protein that are found abundantly in the coelomic fluid and tissue homogeneity. So that's why glycoprotein, GP. So glycoprotein, yes. But these are, they are, protein. As you can see here, 38.6, their molecular weight is measured by the KD. KD is actually extracted by or identified by the apparatus, the biomolecular apparatus, and they are protein. The protein substances, lumbresin PG is a bioactive substance that is with a molecular weight not identified still, but they are short peptide with 39. Look, for example, the other one that is OEP3121 that was first identified from Leucaena fetida, short peptide, and they are protein and glycoprotein in general. GP is to mean G90 means glycoprotein. The scientist, yes, the scientist is first named for the tissue homogenate or tissue extract G90. G90 is simply a glycoprotein. Yes.
Do they also make some lipopeptides?
Yeah, yes, they are cool because they have many amino acid chain. Some of them are very short, and some of them are very longer chain, and they can form this.
I see. We are having a lot of shortage of antibiotics, and we need many new antibiotics. And if we do not have antibiotics by the year 2050, many people may die because of drug resistance. Do you think this earthworm glycolipids and lipopeptides can be used to make antibiotics?
Yeah, look, in one of my paper, we suggested with another co-author that the bioactive substances that are found in the coelomic fluid and the tissue homogeneity should be assayed and identified and genetically engineered to produce them in large amount in industry for use at commercial purposes. Look, in one of the paper that recently published in horticulture, vermicomposting and its derivative against phytopathogenic fungi in the soil. We have, stated this future direction. In future direction, the bioactive substance that are found that I have listed there, there is a genes for them. Every trait is coded by gene. So the gene that code for every of the bioactive substance that are found in coelomic fluid and G90 should be assayed and then amplified. Even genetically engineered, like insulin can be produced by genetically engineered bacteria or any other hormone. Similarly, and this bioactive substance can also be produced in large amounts, can be used as antifungal and antimicrobial activity. Antibiotic also, yes.
Yeah, this will be a new source of antibiotics, and it may help us by the year 2050, and many people may not die because of antibiotic resistance. So we continue work, and this is applied work, and try to patent some antibiotic which is made by these earthworms.
Thank you, sir.
And then we can produce antibiotics, right? To develop an antibiotic, it needs about 15 years. They are not done in 1 year or 2 years. It will require animal study and human study. It takes about 15 years to develop an antibiotic. So you should, focus on the production of a new antibiotic, which will help mankind.
Yes, sir. Thank you, sir.
Thank you so much. Thank you.
Thank you, sir.
Thank you so much, Saranjeet sir, for adding inputs, beautiful insights to the research work of Mr. Gudeta. Yes. And it was wonderful hearing you. And it's always wonderful hearing to you. So, sir always adds some beautiful inputs, wonderful inputs that can be used in the future direction to make the research a more specific, more viable and commercial. So thank you so much, sir. And
Thank you.
if you have any more queries related to it, you can I'll be just sharing the email ID of Gudeta sir, and we can easily go for our queries. And if anybody here have more queries, can just type in the chat box or unmute themselves for the questions and all. So if we have more queries, we can proceed. And if we don't have queries, we can end the session. So I think we don't have more queries. So Gudeta sir, I have a type of like not a query, but a suggestion that yes, we are moving towards an integrated farming source. So we can also use this particular study, as you are saying, like you have utilized vermiwash, vermicompost. You can also move to some integrated sources or organic sources like Bijamrit, Jeevamrit, and formation of some like some of the organic formulations with the help of some integrated farming sources, not purely organic, but yes, we need some integrated sources because integrated farming is in limelight today. So we can also go for studying the patterns, I think, on like conventional farming as well as the upgraded farming sources to make it more viable. We can extend the sources, like apart from vermicompost, vermiwash, we can utilize many sources which are into limelight today. So that can be a particular suggestion from my side. So thank you for the wonderful talk, and we'll be communicating further with you in the like further talks on Sunday Talkies. So thank you so much, Gudeta sir. It was wonderful hearing to you, and we wish you good luck for your further research work.
Thank you, ma'am. Thank you, ma'am.
Thank you, everyone. Thank you.
Bye.
Yes, ma'am.
Yeah, thank you. You please share the mail ID so that we can communicate further.
Sure, ma'am. Sure, ma'am.
Yeah, this is
I'll be sharing it on the group, WhatsApp group.
Yeah, sure,
and also the presentation of Gudeta sir, so that you can easily communicate with him for your further research work.
Because it is really a new source of antibiotics and it can be explored for human clinical pathogens also.
Yes, so, sir. Yes, ma'am.
Sir, definitely we look forward for collaborations also. Really, thank you very much for the talk. Yeah.
Thank you.
Thank you so much. Thank you, everyone. And thank you, Gudeta sir. And we look forward for more collaborations in this particular field and extending it further. Thank you so much. Thank you, Saranjit Singh, sir. Thank you, everyone. Bye-bye. Take care.
Hey, Nitika, thank you because you conducted the session very well.
Thank you so much, sir. Thank you, sir. Thank you, sir. Thank you.